Welcome to Lecture 4, where we will discuss the history of life on Earth as well as the genetics of evolutionary processes. The chemical and physical conditions of the early Earth provided optimal conditions for the formation of life. However, these first initial cellular populations did not arise all at once. Rather, they arose in a stepwise process. The first step in this process involved the formation of small organic molecules like amino acids and nitrogenous bases. These initial small organic molecules were produced abiotically. These organic molecules then joined together to produce more complex macromolecules such as our proteins and nucleic acids. These macromolecules were then packaged into protocells or ancestral cells. These cells had a unique internal environment versus the surrounding environment. This was then followed by the development of self-replicating molecules, such as DNA and RNA, which allowed for heritability and for genetic information to then be passed on from one parent cell to subsequent daughter cells. The Earth formed about 4.6 billion years ago as gases and dust condensed, and ice and rocks bombarded the Earth, generating a large amount of heat as well as water vapor. Volcanic eruptions were present, which created the first atmosphere. This initial atmosphere had very little oxygen, along with high levels of nitrogen gas, oxides, carbon dioxide, methane, ammonia, as well as hydrogen gas. Rapid cooling events during this time allowed for water condensation, which then eventually formed the oceans that we see today. Lightning and UV radiation provided energy sources for the formation of small organic molecules, as mentioned in the slide before. Deep sea vents, hydrothermic and alkaline, also allowed for areas conducive to the formation of these organic molecules. If you remember the Urey-Miller experiment, Urey and Miller were able to demonstrate that the production of small organic molecules were possible simply by mixing the right amount of nitrogen, hydrogen, and oxygen, as well as providing it with an energy source such as an electric shock. Hudd and al. Have furthered those experiments, and they have since demonstrated that nitrogen-containing heterocycles can spontaneously react with ribose 5-phosphate. This can lead to the formation of nucleotides. Furthermore, these nucleotides could form hydrogen bonds with each other. This allows for the formation of a primary RNA-based structure. This recent study has been very important in confirming that nucleotides could indeed be formed abiotically without the presence of a life form. The formation of protocells has also been demonstrated. Vesicles can be produced abiotically that provide the base for protocell formation. Vesicles naturally want to form when lipids are added to water. Because of the hydrophilic and hydrophobic nature of lipids, they naturally want to form a lipid bilayer. In addition, when immersed in water, they also want to form a circular 3D structure, much like a cell. Abiotically, these vesicles can be produced. In addition, they can also exhibit basic life properties such as reproduction, growth, metabolism, and they can also produce a unique internal environment. When considering the complexity of RNA and DNA, it is likely that RNA was the first nucleic acid to be produced. This is because RNA can be used for protein synthesis, or it can also serve as a catalyst for the production of more RNA molecules, thus acting as a ribosome for cell replication. It is also important to remember that natural selection can also act at the molecular level to favor more RNA replication. RNA itself can take on different conformations that leads to more replicates being produced. In addition, small mutations in RNA could also lead to better self-replication mechanisms. This would also be favored by natural selection. It is also important to note that RNA can easily get absorbed into vesicles. Thus, vesicles that contain RNA can self-replicate and also store genetic material. Also remember an important point that RNA can also be used as a template for DNA, thus supporting the idea that RNA was likely the first nucleic acid produced. We typically rely on the fossil record to provide various lines of scientific evidence regarding the origin and the history of life. This fossil evidence can derive in many forms. We can find fossils in sedimentary rock layers or strata, in mineralized organic matter. We can find trace fossils, as well as organisms that are preserved in amber, frozen soil, ice, as well as acid bogs. Radioactive dating is very important in establishing the history of life on Earth. Radiometric dating uses the decay rate of various isotopes to determine the age of fossils and other prehistoric material. Isotopes typically have a rate of decay or half-life that can be used for dating. For instance, we can look at the ratio of carbon-12 to carbon-14 in order to date organisms. Organisms typically stop accumulating carbon upon death. This means that upon death the rate of C12 remains constant, while the rate of C14 decays. However, there is a limitation to this carbon dating as it is only useful for up to 75,000 years in history. The fossil record can also provide evidence on how new features arose over time, and how these features evolved to form new groups of organisms. An example is looking at the evolution of mammals. Mammals originated from a group called synapsids. These synapsids branched from a common tetrapod ancestor that is also shared with reptiles, including dinosaurs and birds. These synapsids showed and demonstrate the development of a hinge bone that joined the upper and lower jaws. However, in the evolution of mammals, we see the differentiation of teeth as well as lower jawbone fusion. These original hinge bones have now become reduced and are now used for hearing purposes. The fossil record can also provide evidence for key life history events. These events include the origin of unicellular life, multicellular life, the colonization of land, as well as various types of extinction and radiation events. The diagram included here includes some of these key events. The first evidence of prokaryotic life forms occurs approximately 3.5 billion years ago. These fossils are commonly called stromatolites. These fossils occur when prokaryotes bind to thin films of sediment together within the rock layers or within the strata. Another important event in the fossil record is termed the oxygen revolution. Though we are dependent upon oxygen, this was not always the case. Oxygen was initially considered to be toxic because it's highly reactive as well as disruptive to chemical and cellular structure. However, chemical analysis of the fossil record shows a drastic increase in atmospheric oxygen that occurred about 2.4 to 2.7 billion years ago. This increase in atmospheric oxygen correlates to an increase in the diversity and success of photosynthetic prokaryotes. These photosynthetic prokaryotes release oxygen as a byproduct of photosynthesis. As these organisms began to diversify and flourish within the environment, the oxygen content of the atmosphere increased dramatically. Once again, natural selection processes then favored organisms that utilized oxygen. As the content of oxygen increased in the atmosphere, so did the natural selection processes on organisms to utilize oxygen instead of being killed by it. As a result, organisms that could utilize oxygen began to flourish within this environment. This drastic change in atmospheric oxygen caused natural selection to cause three major changes noted in the fossil fauna. This included the extinction of many prokaryotic ancestral lines, the survival of anaerobic bacterial species, typically in oxygen-deprived environments, and the adaptations for many prokaryotic organisms to use oxygen. This led to the development of aerobic respiration pathways. Shortly after the oxygen revolution, we note the first eukaryotic cells begin to appear within the fossil record around 1.8 billion years ago. It is hypothesized that these eukaryotic cells arose through the process of endosymbiosis, when a larger prokaryote ancestral cell or host cell ingested a mitochondrial chloroplast-like bacterial cell. Eventually, this bacterial cell became an organelle within the host cell, now termed an endosymbiont. When this occurred multiple times within multiple different cell types, a beneficial relationship occurred. The host cell gained the advantage of using oxygen, which was then considered toxic, or using sunlight as an energy source, and the endosymbiont also gained an advantage by gaining protection and as well as access to nutrients. There is much evidence that has been presented to support the endosymbiotic theory. Some of these lines include the fact that mitochondrial and chloroplast membranes strongly resemble bacterial plasmal membranes. Both of these structures can replicate independently by binary fission, much like bacteria does. In addition, both of these organelles have plasmid-like naked DNA. Both can transcribe and translate their DNA into proteins independently without the need of their host cell, and both contain ribosomes that are similar in structure to bacterial ribosomes. Please refer to the following diagram for a graphical representation of the endosymbiotic theory. We first start off with an ancestral prokaryote with plasmid-like simplified DNA, plasma membrane, as well as the cytoplasm. Through the unfolding of the plasma membrane, we can see the development of organelle-like structures such as the nucleus, nuclear envelope, as well as endoplasmic reticulum. This ancestral host cell would have then engulfed aerobic bacterium, primarily for the purpose of utilizing it as a food source. However, once this bacterium was packaged within a vesicle, it may have been able to persist within the host cell. This would have led to the formation of our ancestral eukaryote, or our ancestral heterotroph, which could utilize oxygen now for aerobic pathways. It is also proposed that endosymbiosis occurred again, with the ancestral eukaryote now also engulfing a photosynthetic bacterium. As a result, this would have led to the production of an ancestral photosynthetic eukaryotic type cell. Shortly after the development of eukaryotic cells, multicellular eukaryotic organisms began to emerge approximately 1.3 billion years ago. As prokaryotic cells diversified and also increased in complexity, so did the compartmentalization and division of cellular labor. This resulted in the formation of complex organelles, that of which we see in today's eukaryotic cells. This increased complexity also allowed for multicellular organisms to also develop. This can now be represented in our present-day algae, plants, fungi, as well as animals. Shortly after the appearance of eukaryotic cells, larger, more complex eukaryotes and more diverse multicellular organisms became more prevalent. We refer to this as the Ediacaran period that occurred approximately 600 million years ago. Prior to this time period, prokaryotes, one-celled eukaryotic cells, and small multicellular organisms dominated. However, during this period, we can see that more complex, soft-bodied multicellular organisms begin to dominate the fossil record, such as the smallest fossil found approximately 560 million years ago. Another important radiation event is called the Cambrian explosion. This refers to the appearance of many of our present-day life forms that occurred approximately 535 million years ago. Prior to this time period, grazers, filter feeders, and scavengers dominated the fossil record. The Cambrian explosion is characterized by what is termed the predator-prey arms race. We see the rapid evolution of predators with many adaptive features that were used to catch and consume prey begin to appear within the organisms in the fossil record. We also see prey with elaborate defensive structures also appear. Thus it appears that predator and prey items were both in an arms evolutionary race. Here we see a trilobite fossil showing very armored defenses that would have prevented them from being preyed upon, as well as the top predator here with elaborate tentacles that would help it catch prey. The Cambrian period is denoted by rapid change in body forms and appearance of many of our present-day organisms, such as sponges, cnidarians, brachiopods, annelids, worms, mollusks, as well as arthropods. Another important event in the history of life on Earth was the colonization of land. Terrestrial plants, fungi, and animals appear in the fossil record approximately 500 million years ago. Prior to this time, life was considered aquatic. There are many adaptations that made it possible for plants and fungi to live, breathe, and reproduce on land. Early adaptations include the presence of a vascular system, as well as defenses against dehydration that would have made it advantageous for that organism to live in a terrestrial type of environment. Late adaptations include the development of true roots as well as leaves. The presence of plants and fungi on land was then followed by the movement of arthropods such as spiders and insects approximately 400 million years ago, and eventually tetrapods that originated from lobe-finned fishes approximately 365 million years ago. The fossil record also contains evidence for various extinction and speciation events. These events can be influenced by the movement of plate tectonics, mass extinction events, as well as adaptive radiation events. The Earth's crust exists in various tectonic plates that can move and reshape the landscape. There can be both short-term and long-term effects from these plate tectonics. Short-term effects include the formation of islands, turbulent areas of tectonic activity that include earthquakes as well as tsunamis. Long-term effects include continental shifts, wide-scale habitat changes, climate changes, as well as allopatric speciation events. This slide here demonstrates the importance of plate tectonics, beginning with the formation of the supercontinent Pangaea, ending with the current state of the landscape that we know today. The other diagram represents the current location of the plates as well as their current movement. Note that the blue arrows represent the plate tectonics shifting away from each other, while the red areas denote high levels of activity from plate tectonics moving towards each other. This is typically exhibited by the creation of fault lines in areas where the plates are moving away from each other. Where they are moving over each other are usually high activities of volcanic activity as well as earthquake activity or shoreline tsunamis. There are 5 mass extinction events that are well documented within the fossil record. These can be denoted by the red arrows pointing upward in the bottom of the diagram. 2 of the most infamous extinction events involve the Permian and Cretaceous extinctions. The Permian extinction led to a loss of 96% of marine life due to volcanic eruptions that resulted from the movement of plate tectonics. The Cretaceous extinction, however, led to the loss of more than 50% of marine and terrestrial life. This was due to the presence of an asteroid as well as the presence of an ice age which followed. These mass extinction events are typically followed by periods of adaptive radiation and speciation. This occurs as new species arise to occupy new habitats and ecological roles. An example of this involves the Hawaiian Islands. Organisms colonize new islands and adapt with high speciation rates. This leads to the formation of many endemic species.